having me. This is a very special moment for me because I am going to talk about hydrothermal vents. So being Scripps is a very special moment. So I'm happy to share it with you and also about my PhD journey. So the link in hydrothermal vents is very important between the ocean space sciences. Since its discovery in the 70s, the efforts to study the deep ocean and these extreme environments has increased a lot in these last years, also because of all the technology and all the improvements that it has been made through study the deep sea. And also, not only it was an outstanding discovery for geophysicists, geologists, also for biology and microbiologists, that we found these extreme ecosystems when we are finding these extremophiles that are high, with a high diversity, not only in microbiology, but also on macrobiology. And in a bigger scope, hydrothermal vents are very important for astrobiology. Bigger scope is the origin of life, which is a very important thing in our planet. And also for looking life elsewhere, also hydrothermal vent systems has been proposed not only as place where these structures have created or originated the molecules for the cells, but also for probably there are similar structures existing in the icy moons. So this is one of the biggest scope that we are studying hydrothermal vents. But in particular, in this topic, we are exploring what is the Gulf of California in Mexico. So if you're will hit the Gulf of California, we are here in Scripps at the north. If you cross the border, here Tijuana and Ensenada, excellent tacos. If you want to stay longer, you can tell me. But for studying the Gulf of California, the deep ocean has been studied for over a decade. And they have found that there is a series of transform faults and spreading centers, which have led to the discovery of hydrothermal vent systems. In particular, we have found, here it is, in Pescadero Basin at the south of the peninsula of the Gulf of California, we have found two hydrothermal vent systems. I'm sorry, they moved. But here, the yellow dot, it's called Auca, and the red dot, it's called Jaigma-Ja'a. Auca was discovered in 2015. And Jaigma were discovered in 2018. These are the hydrothermal vent fields that are at a depth of 3,650 meters depth. This is the deepest known hydrothermal vent system in the Pacific Ocean, which has led us to study them as a very extreme ecosystem. And we have found that the geochemistry of these hydrothermal vents it's very unique. They are made of calcite, and there have been some temperature measurements between the 20 degrees to 100 degrees Celsius around the sediments, but one of the highest hydrothermal vents temperature goes around 300 degrees. So one of the questions that we have, one of the big questions that we have for studying these hydrothermal vent systems is who are the microbes related to the hydrothermal fluids in the sediments of the Auca and Jaigma hydrothermal vent fields, because we don't know a lot of these hydrothermal vent systems. So the first question was, who are them? So for this, an oceanographic expedition was made in 2018 aboard the research vessel Falcor. This research vessel has a ROV called Sebastian, which is the rover that let us submerge, and with a robotic arm, it was able to take some samples of the sediments. So here you can see a little insight about how are the sediments around these hydrothermal vents. All the white stuff that you see around is microbial mat. So we collected around 30 push core sediments, and all these core samples were sectioned on the ship, and it was subsampling with for taking DNA extraction to do pore water analysis. Back in the lab, we made some ion chromatography, DNA amplification of 16S, some sequencing and bioinformatics, and of course some statistical analysis. And so here I'm going to present you some of the results of this. So one of the questions we also have in the first place, it's how are AUCA and Yaigma compared? How are these hydrothermal vent systems related? Because AUCA and Yaigma are like 2 kilometers away. So we wanted to compare how much are they similar in terms of the geochemistry and all the microbiology that we have been finding. So we performed this MDS analysis. So if you see there, we have 3 different locations. AUCA and Yaigma are the hydrothermal vent fields, but we also have another location that is called Uwagyu. I'm sorry for the names. All the names are native words from the region. So that's how we decided, well, scientists decided to name them. These both are hydrothermal vent systems, but Uwagyu is a site between them, but it's a dead hydrothermal vent site, so it's not active anymore. So we use it as an outsider to do this test. So in general terms, you can see that they are very similar, AUCA and Yaigma. And this is the outsider Uwagyu, that it looks not that similar to them. So they are sharing a lot of things, but we don't know what, if it's the geochemistry of this, the microbiology. So we want to go further in to study the microbial composition of both of them and compare them and to see if they are related or what are the differences between them. And one of the biggest surprises that we have is that the microbes are related to the hydrothermal fluids, but between AUCA and Yaigma, the microbes are different. We have different film composition, but the only film they share is thermatogota. So this is a very interesting thing to explore, that we are finding different microbial communities between those hydrothermal vents, but they are sharing one. So this is telling us about the thermophilic and hydrothermal fluids, all the ambiental, all the conditions, the extreme conditions that are around, and also they tell us about all the geochemistry that is happening. It's telling us about it's very complex, I think, all the metabolism that is going on between these sediments around hydrothermal vents. So we are trying to explore furthermore about this group, the thermatogota, that they are extremophiles. They are adapted more than 50 degrees or 80 degrees Celsius. So we are trying to understand if these are like the same families that they are sharing or not. That's one of the next steps for this. But also the implications for astrobiology in terms of hydrothermal vents is that we are still exploring a lot of findings of thermophilic microbial communities in these hydrothermal vent systems to try to understand the limits of life on our Earth, as it is one of the deepest points of the Pacific Ocean. It is telling us a lot of information about that. And also another step for the investigation that is going on, it's applying it to the icy moons with habitability models, where we can try to add these ecological models, and now we have data to try to simulate this. We have geochemistry data, we have microbiology data, so we can try to approach ecological model to the icy moons. If someone in the icy moons wants to collaborate, please let me know. Well, the highlights of these presentations to take away, I want you to to show you some of the hydrothermal vents, how the hydrothermal vent system looks like in Pescadero Basin. But this is the first insight into Jaikmaja, a hydrothermal vent field, and its microbiology. It's the first time that we have seen this. Our findings provide valuable insight into the deep sea biosphere, and that we are learning that there are a lot of information that still need to be processed and to know and to discover. And we have found a diverse range of extremophiles in microbial communities. Right now, I'm just talking about the thermophiles, but we have methane oxidizers, anaerobic microbial organisms. So there are a lot of things going on in the depth. And well, knowing that the Pescadero Basin is the deepest hydrothermal ecosystem in the Pacific Ocean, well, it implies that we are still understanding the limits of life on Earth and beyond. So with this, I want to thank you. Please, if you want to know a little more about the Mexican Astrobiology Society, there is a link. And I am also from the Astrobiology Lab at UNAM, and we are happy to collaborate. Thank you.
Thank you for your talk. I was just curious, do you find many nanoprokaryotes like nanoarchaea or like candidate phyla radiation bacteria, or do you just filter them out in your analyses somehow, or like are there any interesting patterns?
Well, right now, we are finding also archaea and bacteria, but that's like the farther that we have. Like now, there is a lot still of information to show, but there's a lot of things going on.
Yeah, I just remember like looking through various papers. There are a lot of them, a lot of those like nano-sized clades present in the hydrothermal vents.
Like nanoarchaea, or
yeah, like nanoarchaea or
yes, we have a nanoarchaea is around, and AbGradCon is also in there. Yes.
Yes, sorry. Just kind of curious. I'm a physics kind of background person from UCSD, and I was wondering about temperature management. So, like, you know, above certain temperatures, like you're going to get denatured proteins and stuff like that. And I was wondering if there's extremophiles that do that cool themselves somehow, or anything like that. Sorry, just totally ignorant.
Yes, actually, well, right now I'm talking again as with thermophiles, but actually here in the hydrothermal vents, well, not the hydrothermal vents, around the hydrothermal vents, all the sediments, there are different changes in temperature. So right now I am talking about the highest ones that we have measured, but if you go farther, the hydrothermal vents, it drops to 2 degrees centigrade. So we are studying, it's able to study different microbial communities adapted to high temperatures, but also to coldest places around.
Any other questions for Manet? Oh, Kelly?
Hey, really awesome talk. This is really cool. I worked with hydrothermal vents as an undergrad, so this is like really nostalgic for me. I'm curious if anyone in your group or anyone part of the cruise is doing, and maybe Karen Lloyd wouldn't like this, but is doing any culturing from these environments.
Yes.
Like specifically, I'm thinking at high pressure, because these are some of the like deeper vents in the Pacific, or the deepest vents in the Pacific, I think you said. And so I'm just curious if anyone's doing culturing and if they're including pressure in that.
Yes, actually, one of the here in one of the collaborations that we have is in Caltech, in Victoria Orphan's lab. Yes, the group are making some culture experiments with some of the different sediment cores in different sites. So they are trying to do that. And they have also published one paper about culturing archaea, I think. So they are doing it, but of course, the challenging things about the pressure. So they have like this tank to try to approach that. Yes, they are trying to do cultural techniques for this.
Any questions from up top? Any questions here?
I thank you for the talk. I have a very trivial question. We've been having issues with sediment samples in our lab, freaking out how not to contaminate them or not being sure if they are contaminated when we are taking the samples. I wonder how you control for that.
Yeah, that's a good question. So when we are working on the ship back in the lab, there is a lot of possibilities that it got contaminated. We're trying to be as quick as possible and in clean spaces, but we actually, where we are working with sediment cores, it's mud. So it gets really mud all around. We try to clean, but we just try to be as quick as possible. One of the things that we rely on is the bioinformatic analysis. So in the bioinformatic analysis, when we are processing all the 16S data, the sequences, we are filtering them. So we have like we have also for the 16S or the DNA extraction some blanks and some control samples. So we tried like if there is a sequence in the blank on the control, we delete them. So we try to filter and to make the cleanest sequence possible. But yes, of course there is some contamination, and we also like sometimes we see like, oh, that's not from the deep sea, but we try to see the sequences and try to clean them. That's why. Mm-hmm.
Any last questions? Okay, thank you very much, Manet.
Thank you.